These viruses have evolved the ability to hide themselves from the human immune system by covering themselves in a sugary shield. And what that means is that when the virus is inside the body, the human immune system doesn't see the virus as a foreign agent. My name is Rommie Amaro, and I'm a professor of chemistry and biochemistry at the University of California, San Diego. Up until 2020, my team has been trying to push the limits of molecular simulation from smaller systems to beyond scales that anyone has ever been able to achieve. And we had been performing these simulations on the influenza virus. We had actually just recently published our influenza study that had actually lasted about 6 to 7 years. The same week that that work went to press, there were a number of things happening worldwide that were very important. Even though cases of COVID-19 hadn't really come too much into the US yet, we had maybe just about a dozen circulating cases that we knew about. We knew that it had left mainland China, and it was circulating already in the Lombardy region of Italy. And so things had already started to look pretty grim there for worldwide transmission. That sort of got our attention. And then that same week, another important piece of data came forward, and that was from the group of Jason McClellan and his collaborators at the National Institutes of Health. They actually published the first SARS CoV-2 spike protein structure. And as soon as we saw that structure come to light, we picked it up and started running with it immediately. I'll never forget walking down to my postdoc's office and saying, you know, folks, I think we need to do something with this SARS CoV-2 virus, and the data is now out there. And I walked back and I picked up the phone and I called Dan Stanzione, who's the director of the Texas Advanced Compute Center. And at that time, I knew that if we were going to do something serious for SARS CoV-2, that we needed the compute time to be able to do that. They were very generous in giving us essentially unfettered access to the Frontera supercomputer, which formed the basis for much of the successful work that we've been able to accomplish in SARS CoV-2 research. One of the first things that scientists wanted to do was to figure out what it looked like. And you know, it's important to know what your enemy looks like in some sense, right? Because you want to understand how it's working exactly. And so scientists have developed ways to take pictures of the virus and the important parts of the viral machinery. Most folks are familiar by now with what the virus actually looks like. It sort of looks like a golf ball with spikes sticking out of it. Thanks to the data from Jason McClellan and David Wiesler and other co-workers around the world, these structural biologists, they were able to use a technique called Cryoelectron Microscopy, which gives near atomic resolution of what the spike protein looks like. But one of their big challenges, or one of our challenges as scientists, is that as powerful as this technique is, it only allows us to take essentially static snapshots, like Polaroids, of these biological systems. There are some things that the method can't see. So it gave us a picture of the spike protein, but there were parts of it that were missing, essentially also because they moved too much. So the researchers in my group worked to rebuild what we call a full or complete spike protein model. The other part that the structural biologists can't see relates to a very important aspect of viral proteins, and that's what we call the Glycan Shield. Basically, human cells are covered in this sort of glycan coating or sugar coating. And so viruses have evolved the ability to essentially cover themselves with the same type of sugar molecules that are found on our host cells. And by doing so, that sugary coating acts as a disguise or a mask from our human immune system. The catch is that the structural biologists can't see those sugars. So this is a place where molecular simulation, together with high-performance computing, has a lot to give. We rebuilt not only the missing bits of the protein, but all of the sugars that are decorating the structure also. And then not only do we see that what they look like sort of statically, but then we're using these all-atom molecular dynamic simulations to predictively animate the motion of the sugars and the spike protein. And this is so important because if you want to understand how a system works, especially a highly dynamic system like the spike protein in these viruses, one needs to be able to see how it's moving, how it's interacting with other parts of the host cell, like the receptors on the human host cell that need to come into contact to precipitate infection, and so forth. And so the only way for us to do that right now, currently, for scientists to explore the dynamic motion of these molecular machines with the detail that we need to understand, for example, what those sugar molecules are doing, we need to use high-performance computing. So when researchers looked at the pictures that had been made available by the cryoelectron microscopists, what they saw was the spike protein in two conformations, a closed conformation and an open conformation, or a down conformation and an up conformation. And this is how they describe them. But when we actually included the sugar molecules, and when we actually let them move around and we explored the dynamics of the system through molecular simulation, what we found was that those two conformations of the spike protein were really important functionally. In the closed conformation of the spike protein, the entire protein was completely masked or covered over or disguised with the sugary shield. But in that up conformation that they found, what we could see so clearly when we actually simulated the effects of the glycans was that the spike protein was revealing a very important part of itself. Basically, it revealed the weaponry that was needed to actually attack and infect the human cell. As it gets close to the host cell, it actually lifts up out of this sugary shield. It actually changes, it shapeshifts, if you will, and exposes the part of the spike protein that actually makes contact with the host cell. That really told us what we needed to know about, or one of the things we needed to know about the function of the virus itself. One of the most exciting things about our work was that we showed the world for the first time what this fully decorated spike protein actually looked like, so the Glycan Shield of the spike. And that itself was a really sort of important understanding for scientists. But we also discovered something else. And that was when we looked at the spike protein, when we really looked carefully at that up structure, that attack structure, what we saw was that there were two sugar molecules in particular, two sugar locations that basically were propping up that spike conformation in its attack mode conformation. And so we performed experiments both in the computer as well as our collaborators, Jason McClellan and his group, tested the same experiments actually in the lab. We actually discovered that there were two glycans that if they were removed, that up conformation collapsed. And essentially it prevents the virus from being as effective to actually infect human cells. This finding was really only possible through the molecular simulations that we carried out on Frontera. And not only was that interesting for SARS CoV-2 research, but it was also the first time ever for any viral protein or any system that the glycans themselves were shown to be part of the infection mechanism. And so this presented a real sort of game changer finding for the scientific community in terms of the roles of glycans in biological systems. So in our first set of findings, we were really focused on understanding what those two different structures really looked like with their full sugary shield. But one of the things that we were next interested to understand is actually how the spike protein moves from that down conformation to the up conformation. And in order to do that, it's much more computationally intensive and required us to utilize a different part of the Texas Advanced Compute Center and actually take over many hundreds of GPUs on their Longhorn machine. And so there, what we were able to do is use what we call an enhanced sampling molecular dynamics approach called weighted ensemble simulations, in order to really study what we call that functional transition from the down to the open state. And what was exciting about the findings of this particular work, not only from a computational standpoint, because it brought many challenges in terms of running out to many different GPU nodes and dealing with an enormous set of data, reconstruction and so forth that resulted from that. But scientifically, what was so interesting was that it allowed us to discover yet another new glycan. This particular glycan didn't strike any of us as particularly interesting. But when we actually did the hard work and all the simulation time to explore that functional transition, we discovered a glycan that actually helped to lift up that part of the spike into the up conformation. And so this was a finding that, again, we made a prediction that we tested using simulations, and then our counterparts, our experimental collaborators, Jason McClellan and co-workers, actually did more experiments in the lab to test our hypothesis. And what they found was that this glycan that we discovered using this even more intensive computational approach actually played an even larger role in the infection process than the previous two that we had discovered. And so this was another important finding that will have ramifications for understanding how this virus actually works and infects human cells. So after we studied the single glycoprotein, one of the things that we were really interested to do was to actually understand how that spike molecule actually moved and behaved when it was in the whole virus. And so why we care about that is because the other molecules that are bumping up into the spike protein or that are next to the spike protein or on the host cell that the virus is binding to, they will all affect the movements and the dynamics and the function or the behavior of a single spike protein. So we viewed this as an important next step in understanding the virus itself, was to move beyond just the single glycoprotein and into these whole virus simulations. And to do this really, again, sort of pushes the boundaries of molecular simulation and high performance computing. Because the spike protein, whereas it has about 2 million atoms, when we actually reconstruct the entire virus, it has over 300 million atoms. And so this presents an enormous challenge for computing at scale. There were about 30 different researchers at, I think, 6 different institutions, and where we used not only the tech machine Frontera, but we also used a number of other platforms as well to perform multi-scale simulations of this whole big virus. Trying to visualize these large systems in a way that the researchers can actually interact with them in real time is very challenging and something that we're continuing to work on with these great teams. And then from there is really where the new discoveries are going to be made. We realized that this challenge of the virus, of COVID-19, was unlike anything that any of us had ever faced before. In order to respond to this new challenge, we needed to do things differently than how we normally operate. And so I was really proud of our community. We came together pretty early and agreed on a fundamental set of community principles that would sort of guide our research in this space. And one of the reasons that it was so successful, because we had so much great input from collaborators all around the world in terms of the models that we were actually building and the analyses of our simulations. They have been, in fact, looked at and worked on by researchers in the pharmaceutical companies, as well as in other academic groups. Our simulations have shown the world what the sugary coating actually looks like, and also how it's used to hide the virus from the human immune system. And when we know how the virus is hiding, it gives us a better chance to try to understand where the vulnerabilities in the shield or in the virus are. One can then think about designing small molecules or neutralizing antibodies that could take advantage of these sorts of chinks in the armor, if you will, these vulnerabilities. Making things like vaccines actually detect the virus inside the body. We can use these new advances, new algorithms, new architectures to bring forward information that otherwise would be completely hidden, but that can be really tremendously valuable in the fight against disease.